Hello, everyone, and welcome to today's live broadcast, The Use of 3D Models in Hepatic Biology. I'm Christy Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Thermo Fisher Scientific. For more information on our sponsor, please visit thermofisher.com. Now let's get started. I would like to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. Also, please notice you can share this webinar on your personal social media. Just click on the Social Sharing tab to let your friends and colleagues know about today's live event. If you have any trouble seeing or hearing the presentation, click on the Support tab found at the top right of your presentation window, or use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I'd now like to introduce today's presenters, Dr. David Kuninger, Director and Group Leader at Thermo Fisher Scientific, Julia Tritapoe, R&D Scientist, Cell Biology at Thermo Fisher Scientific, and also joining us today is Michael Millett, Scientist 1, Cell Biology at Thermo Fisher Scientific. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. David, you may now begin your presentation.
Yes, thank you for that introduction, Christy. Hello, my name is David Kuninger. I'm a Director of R&D at Thermo Fisher Scientific, and it's my pleasure, along with two of my colleagues, to present this webinar on in vitro 3D spheroid culture models using human primary hepatocytes, HepaRG cells, and primary animal hepatocytes. Okay, so I thought I'd start with some broad terms and definitions as we sort of get more specific into the hepatic models. So, broadly, what is a 3D model? It can mean a number of different things. This illustration is just meant to kind of convey the broadness in which folks are often will term or consider various in vitro models as 3D. And so just going from left to right, we've got traditional monolayer, and then moving more towards the right, we have more classic co-culture models that can be used with transwell inserts. These have been around for decades, and then getting more into what I think we more properly or more timely definitions of 3D models, and that's the spheroids and organoids, and then moving into more complex systems such as organs on a chip and bioprinting. Really the key rationale for building these more complex systems is to drive more physiologically relevant responses out of the different cell models, and I think this will be illustrated with the hepatic models that we'll be discussing in detail. So the next slide gets into the weeds a bit more with the models, and this is really parsing definitions between spheroids and organoids, and I think I tend to think of these as somewhat loosely defined. Sort of working definitions are shown here. So with the spheroid, is a 3D aggregate composed of one or more cell types that grow and proliferate, although not all models necessarily will proliferate. Coming back to this, but also often exhibit enhanced physiological responses. And may or may not go more complex processes of self-organization and differentiation. In contrast, organoids are typically derived from proliferating populations that are capable of self-organizing. and these are often taken from, are initiated from either pluripotent stem cells or tissue-derived progenitors that are capable of both proliferation and then more complex, formation of more complex structures and different cell types. And also tends to mimic the in vivo architecture of the tissues more often. Below is just a number of cell sources that can be derived to initiate both spheroid or organoid cultures. With that, I will keep rolling. Okay, so getting into our primary hepatic human spheroids specifically. So why, again, why would researchers, scientists be interested in using 3D hepatic cultures? You can read through the slides here, the table here. The bottom line, I think there's really a couple key features. One is the physiological responsiveness of hepatocytes in 3D is much more robust than 2D. The longevity, most of you listening may know this or may not, but typically, at least human primary hepatocytes, when plated ex vivo, don't last very long. You can, a week, if they plate at all, and maintaining the cultures at high viability and high levels of confluence and activity for a week is a challenge. So this provides a fairly narrow window and fairly challenging conditions to be able to do this consistently. In contrast, the 3D spheroids of these hepatocytes, so these would be non-proliferative human primary hepatocytes that are cultured in a way that promotes their self-aggregation. So in this case, with the primary human heps and the animal models that Julia will speak to later, these are non-proliferative cell types, and we're using culture conditions that promote the self-aggregation of these cells into spheroid structures. So non-proliferative for the primary heps, but once they form these spheroids, they maintain viability and functionality for weeks. And on a per cell basis are much more active. So you get much longer term cultures and much higher activity. In some ways, the complexity, once one has a lot of cells that have been screened or they're capable of forming spheroids, it's actually not that difficult to consistently create spheroids in these in the Sphera 96 well plates is the format that we've been using fairly extensively. It's actually fairly straightforward to do, unlike plating monolayer cultures of primary human hepatocytes, which can be challenging to do consistently. Some of the other, again, from a biological standpoint, the longer culture windows enable the study of low turnover compounds, the ability to recapitulate more complex pathophysiological disease type processes, and co-cultures becomes attractive. A paper I will reference a little bit later also suggests or indicates the predictivity certainly with respect to hepatotoxicity is vastly improved in the 3D cultures versus traditional 2D from the same source. So a number of really key benefits in using 3D culture in this type of 3D culture system. Okay, so I'm going to jump right into the workflow with our primary human hepatocyte spheroid system, or kit. This is actually a kit we put together, launched last year, about 9-10 months ago, that consists of cryopreserved human hepatocytes that have been pre-qualified, screened for their ability to robustly form spheroids. The thaw, plating, and maintenance media required, and in addition, the Nunclon Sphera 96-well plates, so to enable the spheroid formation and maintenance. The workflow shown at the top, it's again pretty straightforward. You just thaw, count, and plate. As the cartoon indicates, the cells initially, are individual, singularized, and then they, in the ultra-low adherent, round bottom Nunclon Sphera plates, the cells will self-aggregate and form these spheroids over time. And we typically will plate and leave them unperturbed until day 5, where we do a media change and inspect the cells for the quality, morphologically, the quality of spheroid formation. And at that point is when we typically start to do experiments with these cells. And this is in contrast to the platable version of these cells. By day 5, they're really exiting their usable window for the most part. The picture below, the photomicrographs just show what this looks like when you're peering under the microscope. And again, I think just the box on the right, well, you see a kind of a blow up on the upper right of what these spheroids look like at a higher mag, and then just the components of the kit shown in the box. Oh, yes, and a tip. So as we've been working more and more with these 3D models, the hepatic 3D models and others, frankly, we've been learning a lot about kind of what works, what doesn't work, and what are things that we have to adapt along the way. So we've tried to sprinkle in a few helpful tips that we've learned to either help with the workflow or perhaps give you more reliable results with different types of downstream assays. And in this case, our tip is if you're using the multi-well plates, consistently doing media changes and or washes can be challenging with a single spheroid in there. So we really like the an automated microplate washer, the well washer Urshas, one we use routinely, really facilitates the ease of use, the speed, and I think probably helps with consistency of the overall cultures using a system like this. Okay, this diagram, there are other commercial options for pre-qualified lots of cryopreserved human hepatocytes. And this slide is just meant to indicate that our system on the left is when we spent a lot of time in both the cell optimization and workflow optimization, and just pulling the components together to really simplify it. And as I mentioned on the earlier slide, we basically thaw, count, and seed, and then leave the plates alone for the human hepatocytes until day 5, until the first media change. Excuse me. Other commercially available systems require substantially more manipulation of the cells, pulling them in and out of the incubator multiple times to do this. So just increased risk and more work, but just a comparison of the workflows. Okay, so this slide is just going to walk through some of the basic parameters that we've sort of did in our routine optimization across multiple lots, and as we were trying to get the product and the protocol dialed in. Essentially, what the slide, so you have a collection of photomicrographs on the left that go, so if you look at the numbers at the bottom, 750 to 7,500, that's initial, that's cells that were seeded in a single well of the 96-well Nunclon Sphera plate. And so we're increasing cell numbers from left to right. These images are taken at day 5 following the workflow I showed earlier. And what we see is a fairly consistent, so these are just replicates that you see in the column form. And as we go across the rows, increasing size as we increase initial seeding density. We can quantitate that by analyzing the images and calculate the volume. So we're able to, as you can see on the left, and also see it the quantitation on the right, we're able to increase the size of the spheroids by starting with more cells in the initial seeding within this range. And as one would hope, I guess, or expect to see, that spheroid volume is proportional to the number of cells per well. So we feel like, for the most part, with these, the vast majority of the cells that are seeded, viable cells that are seeded, are included in the formation of these spheroids. This data is consistent with that. Okay, so kind of a look inside these spheroids. So we've got two types of staining here. These are cells that, when fixed and stained, the top, and again, if you look at the numbers at the lower right of each of the images, represent the starting cell number at the time of spheroid seeding. So what we see is, again, sort of an increase in size as we're going from the lower to higher number of cells. But also, if we look at the top panel, this is hematoxylin and eosin staining, H&E staining of sections, of cryosections of the spheroids. So we see a fairly uniform distribution of cells. So these are solid masses of cells that are fairly uniformly distributed. These are just hepatocytes. So this is really a monoculture. The lower row shows tunnel staining of sections as indicated, and here, so we're looking for tunnel as an indicator of precursor of cell death, and what we're really looking for was, can we identify hypoxic or necrotic zones in these spheres of any size? So really looking for a concentration of signal within the core, the center of these, and certainly within the size range and the culture conditions and duration that we've been typically doing, we have yet to consistently identify sort of necrotic cores in any of the spheroids. Okay, so let's see, how do they work? So now we're looking at gene expression. In this case, we're comparing relative to 2D cultures. So the same lots of same cells, either cultured in 2D at day 5, or in 3D at the indicated times here, and we're looking out 1 to 4 weeks out. We've got 3 different lots. So if you, maybe it's a little bit hard to read, but the HU ... those are all different, 3 different lots of cells that we're looking at in parallel. This is RT-PCR, qPCR analysis of different CYPs or transporter, a specific transporter, and albumin. So just starting from left to right at the top, we're looking at CYP3A expression, mRNA levels relative to the blue bar, which you probably can't see because the levels are so low at day 5, relative to, those are all set at 1 also because we're normalizing. But so we see enhanced CYP3A4 expression in all the donors, although we do know there's donor-to-donor variability. Similarly, CYP2D6 expression is substantially elevated in all 3 of the spheroid lots, relative to the same cells plated in 2D. The MDR1 transporter, similarly, high levels in all of the spheroid cultures, relative to 2D, and same with albumin in this case. So, again, consistent with what we would expect to see is enhanced expression of these key transporters that are at the basis of some of the fundamental physiological actions that are relevant for many folks with these types of cells. Okay, so a little more functional characterization. In this case, we're looking at we're looking at the formation of bile ducts via CFDA staining on the upper left panels. So what you can see is the top. We're showing the top row is primary human hepatic spheroids. The lower row is hepatic cell line, HepG2, which does not form bile ducts, bile caniculate. So that's our negative control for CFDA staining. DAPI just to show spheroids are there. And then you can see the co-staining in the merged image. So this is something we fairly consistently see with the spheroids. If we're looking at activity, the figure in the upper right, we're looking at activity of the specific activity for the CYP3A4 transporter in the 3D cultures, substantially higher. I would note, and I think this is a somewhat important point, that we're looking at a per cell or per fixed number of cells at this point. So the spheroids, while they have a relatively small number of cells, are very active. So again, the activity is very high. There may be certain applications where additional biomass is going to be required. You may need more cells than might typically be found in a single spheroid. So this can be addressed by pooling. This is something that we do, and I'll come back to this a little bit later. But this is a common theme that we see with the spheroid cultures across all the models that we'll be discussing is higher metabolic activity relative to the cells cultured in the same cells cultured in 2D. And just below that, we see improved albumin secretion in the 3D cultures relative to the 2D. And again, this is normalized on a per cell basis. Okay, and so we're looking at increased metabolic. So in this case, we're comparing metabolism of 6 compounds across 2 lots of hepatocytes that are cultured in either 2D or 3D. And you can see, it's maybe a little bit hard to see the compounds. They're at the top of each graph. What we find across the board, I should say across the board, with 5 of the 6 compounds that we looked at, were increased metabolic activity with the 3D cultures relative to their 2D counterparts. So this was consistent across the board, except for the hydroxy propione. Sorry about that. And I guess this was a reproducible result. We're not entirely sure why one donor of 2D scored so high here, but otherwise we see consistently higher metabolic activities with the lots in spheroid culture versus 2D. And I just wanted to highlight this. This is actually work from a paper from a couple of years ago from Will Proctor and Allison Foster. And what they were doing was trying to systematically look at whether what they called hepatic microtissues, these are actually hepatic spheroids, or again, I think our definitions of spheroids or 3D structures. We should probably be generous in how we're using it. But in this case, they're calling their hepatic spheroids liver microtissues. They were looking at the predictivity of these these cultures to identify known hepatic toxicants that are known to induce, drug-induced liver injury, DILI. And so essentially they took 110 compounds and screened these of known toxicological profiles and said how well do the 3D cultures predict versus 2D, and it was pretty impressive actually. The microtissues, the spheroids, showed increased sensitivity, increased predictivity, and the authors also suggested that that these models might be useful in identifying biomarkers related to, for biomarker mining related to hepatic injury. So again, a fairly substantive work indicating the sensitivity and specificity of these 3D cultures relative to the same cells in 2D. Okay, so with that, I'll wrap up the conclusions here of this section. Yeah, so we have our Gibco primary human hepatocyte kit. The SKU is there. It's really everything you need to get started in 3D human hepatic culture. Comes with the Nunclon Sphera plates, plating, media and supplements, and the 96-well plates. So as well as we have fairly extensive guidance and application notes, some of which I think Julia will touch on at the end, web-based resources to aid in getting up and running with that. Yeah, I think we'll demonstrate at least in part that the hepatic spheroid cultures, the human hepatic spheroid cultures are capable, are functionally viable at least 4 weeks. Substantially further than their 2D counterparts. Yeah, again, much lower numbers of cells are required for equivalent activities. So again, this may enable these fairly precious and relatively expensive cells to be used more effectively and hopefully to drive better results. Yeah, and again, we see better activity, higher gene expression levels in the 3D cultures as compared to 2D. Okay, and with that, I am going to hand you off to my colleague, Michael, who's going to go over our HepaRG spheroid work.
All right, thank you, David. So as he said, I'm going to talk today a little bit about some of the HepaRG 3D hepatic spheroid work I've been doing. So for those of you who aren't familiar, HepaRG is a terminally differentiated hepatic cell line, and it's derived from a human hepatic progenitor cell line. HepaRG is comprised of two different cell types, 50% hepatocyte-like cells and 50% cholangiocyte or biliary-like cells. And the top left, we have a liver cross-section, and those cells that make up the bile duct are the cholangiocytes, and then the surrounding hepatocytes are the two cell types that compose HepaRG. One thing that really separates HepaRG cells from other hepatic cell lines is that they retain most of the characteristics of primary human hepatocytes. So most of the enzymes and pathways that people use primary hepatocytes for are retained in HepaRG cells. Two advantages of HepaRG over primary human hepatocytes are they are much more cost-effective and cheaper for the customer, and they lack the donor or lot-to-lot variability that plagues primary hepatocyte work. So in the bottom left, this is just a 10x photo of a 2D HepaRG monolayer. You can see two distinct cell morphologies in there, one of them being the binucleated cell that is characteristic of primary hepatocytes. So when I started this work, we were getting a lot of customer inquiries as to if these cells could be used in 3D, and we were seeing a lot of recent literature about these cells being used in 3D. But I stood out only having an established 2D workflow, which is very simple. You just thaw the cells and plate them onto a collagen one coated plate, and then days 6 through 10, you can assay them for various purposes. So I first set out to see if it was even possible to form HepaRG spheroids. In the top left, you can see the workflow. All you do is you thaw the cells and seed 1,000 of them per well of a Nunclon Sphera super low attachment U-bottom 96-well plate. And by day 2, the spheroids have mostly formed, but by day 3, they're fully formed with a really nice outer outline that you can see here. And we'll actually get into it later. That's actually when they're the most functional. In the right-hand graph, you can see it's a little bit of a teaser of the data I'm about to get into, but just overall, cells are much more active in a 3D format compared to a 2D format. This graph goes over a cytochrome P450 enzyme activity, CYP3A4 specifically, and you can see in a 3D spheroid format in a Nunclon Sphera plate, the CYP3A4 activity is significantly higher than even 2D induced monolayers when you induce the cytochrome P450 enzyme activity. This left bar is just the basal 3D activity. So moving on, I characterized the 3D protocol based on a 10-day time course of enzyme activity in the cells. This was also how the 2D protocol was established. This left graph, you can see it's comparing the basal CYP3A4 activity over a 10-day time course of 3D spheroids versus 2D monolayers. The two main takeaways are that at all time points, your activity is significantly higher in 3D compared to 2D. And the second main takeaway would be that actually on day 3, cells reach peak enzyme activity in a 3D format compared to a 2D monolayer. We have to wait through days 6 through 10 to get peak enzyme activity. And so you can assay your cells actually much quicker in a 3D format compared to a 2D format. Moving over to these right 3 graphs, I just wanted to show that the trend of significantly increased cytochrome P450 enzyme activity held across different CYP enzymes. So this leftmost one is CYP1A2, and then to the right of that is CYP2C9, and then here's pulled from the graph on the left, CYP3A4. So it holds true across all the cytochrome P450 enzymes that we have looked at so far, that there is significantly higher activity in 3D versus 2D. That's orders of magnitude higher. So moving on, this is just some quick histology staining we did. The left graph or chart is of an H&E staining of day 14 HepaRG spheroids. A lot of the literature states, depending on what format you grow these cells in, the two cell types, the cholangiocytes and the hepatocytes, one will form a shell and the other will form the inner ball of cells. And this little tiny cutout on the bottom right, you can see that it does look like one cell type does indeed form the outer shell, and the other cell types are in the center. One step we're currently doing is just antibody staining so we can verify which cell type is which. Over to the right, we have a periodic acid shift staining. So the top 2 photos are of a day 3 HepaRG spheroid, and the bottom 2 photos are of a day 14 HepaRG spheroid. The left side is not digested, and the right side has the glycogen digest. So the main takeaway from this would be that it seems the cells seem to store more glycogen over time. So the functionality seems to increase for some applications over time, the longer you let these HepaRG spheroids grow. Okay, so I want to make sure that there was a way to scale up these HepaRG spheroids. This left photo is of day 3 HepaRG orbital shaker spheroids, and these were created by seeding 3 million cells per well of a Nunclon Sphera 6-well flat bottom plate and 3 mils of media per well. You set them on an orbital shaker set to 85 RPMs, and on day 3 you have hundreds, if not thousands, of spheroids per well. These right 2 graphs are just some quick size analysis we did of these orbital shaker spheroids. Unlike the sphere plates, they don't really have a confined space to grow in, so I was expecting them to be much less consistent in size. But as you can see from our graph, even in this orbital shaker format, they're surprisingly consistent, so I was really happy with that. So just to sum up some of the conclusions for our HepaRG 3D work is that HepaRG cells can easily form viable 3D spheroids using Nunclon Sphera super low attachment 96-well U-bottom plates, or on a Nunclon Sphera super low attachment flat bottom 6-well plate placed on an orbital shaker. HepaRG spheroids have basal cytochrome P450, CYP3A4, 1A2, 2C9 were some of the ones we looked at. That's order of magnitudes higher than 2D monolayer culture, and that is both induced and uninduced in 2D. The 3D spheroids have peak basal activity on day 3 as opposed to day 6 through 10, so you can actually assay your cells much quicker when using a 3D format. HepaRG spheroids also store glycogen, suggesting that they're functional, and we can see a bit of spatial organization when looking at these cells in 3D, where it seems one cell type goes towards the outer edges and the others goes towards the inner ball of the cells. So those are all the conclusions I have for this section. With that, I'll pass it off to Julia Tritapoe. She is a R&D scientist with us in Frederick, Maryland as well.
Okay, thank you, Michael, for that introduction. As Michael alluded to, I'll be discussing our preliminary work around our animal hepatic spheroids today. So on this slide, I'm just showing our simple workflow to form animal hepatic spheroids. So very similar to forming human hepatic spheroids. On the first day, you thaw, count, and seed. And then, for the most part, the cultures are left undisturbed for 5 days. At that time, there's a media exchange, and then during week 1, you can do your assays. With the exception that with dog hepatic spheroid formation, this does require on the initial day, day 0 in this cartoon, a longer and harder centrifugation speed, and then subsequent centrifugations on days 1 and 2. And that's only for the dog hepatic spheroids. For the rodent, it's just as human, where you thaw, count, seed, and then undisturbed for 5 days, and then you do the media change. The photomicrographs I'm showing here are just showing, for all of the different species, different cell numbers seeded per well, starting with 375 on the left and going to 3,000 cells per well. Looking at the formation and functionality of dog hepatic spheroids, so in the top left panel, again, looking at optimizing variable cell numbers that we're seeding here. So starting on the left, 375 cells up to 3,000 cells. And you can see there's consistency of spheroid formation among these different seeding densities that we tried. On the top right panel, looking at viability as indicated by ATP synthesis of these dog hepatic spheroids, you can see that they are viable for 4 weeks, which is a huge improvement over the same cells in 2D culture. Which survive typically in 5 days. And then looking at the bottom left-hand panel, this is looking at gene expression in the 3D dog hepatic spheroids. And the first bar on any of the genes we looked at, so we looked at 3 here. We looked at albumin, CYP1A1, and CYP2B6. And the first bar is showing the 2D gene expression, so the same cells plated in 2D. And so these graphs are looking at gene expression as compared to those same cells plated in 2D. And you can see for 2 of the genes, albumin and CYP2B6, we see increased gene expression relative to 2D cultures. And then interestingly, for CYP1A1, while there are transcripts present, they are actually lower than the 2D culture. And this was an interesting finding because if you look at the bottom right-hand panel, the CYP1A1 activity we're looking at in these dog hepatic spheroid cultures, it's actually higher than what we see in the 2D culture. So we can't exactly explain why the transcript levels are lower, yet the activity is higher, but it is what we've observed so far with these dog hepatic spheroids. Moving on to formation of the rodent hepatic spheroids. So on the left-hand side, I have photomicrographs of the rat and mouse hepatic spheroids. Again, we're optimizing the cell number per well here, starting at 375 cells and titrating that up to 3,000 cells per well. And something I will mention with the rodent hepatic spheroids is that we've observed consistently that we find irregular spheroid shapes if you seed higher than 3,000 cells per well. And you can start to see some of these irregular shapes in the rat hepatic spheroids even at 3,000 cells per well. And then on the right panel, looking at viability as indicated by ATP synthesis, you can see that while the rodent hepatic spheroids are viable out to 4 weeks, vitality is decreasing throughout those 4 weeks. This is looking at gene expression in the rodent hepatic spheroids. For rat, on the left-hand side, we looked at albumin and CYP2D4 gene expression, and much like dog that I talked about a few slides earlier, these these are gene expression levels as compared to the same cells plated in 2D. So for rat, we see increased gene expression for albumin and CYP2D4. And then for mouse, on the right-hand side, we looked at albumin gene expression, which we see is higher than 2D, and this peaks at week 2 and then declines after that. And some of the variability that we see in this gene expression data could be due to a low sample size. So this is a single lot that we're looking at here. So we have more work to do in this area, testing more lots for gene expression. This is looking at elevated albumin production in the rodent hepatic spheroids. So rat is on the left-hand side and mouse is on the right-hand side. And what we're looking at here is albumin secretion as compared to the same cells plated in 2D. Day 5 for day 5 2D for the rat hepatocytes, and then for 2D, it's day 3 for the mouse. In any case, we see increased albumin secretion for the 3D models here. So just to have some brief conclusions for this section, for primary dog, mouse, and rat hepatocytes, we've shown we can easily assemble these into 3D cultures within 5 days using the Nunclon Sphera low attachment U-bottom 96-well plate. All of the animal hepatocyte lots that we've tested thus far have shown they're capable of forming spheroids. For dog, we've tested 2 unique lots, for mouse 4 lots, and for rat 3 lots. As I mentioned in an earlier slide, the dog hepatic spheroid formation does require a harder and longer centrifugation speed on the day of seeding, and then also additional centrifugations on days 1 and 2 for optimal results of forming spheroids. And I think this is an important point to think about, as these additional steps might be useful in other lots that are difficult to form spheroids. And then data we're not showing, what we found is that there's multiple assays or workflows that have been designed for 2D that we found require modification to be used for 3D applications. So this is definitely something to keep in mind across all species as you're doing more and more work in the 3D area. We do have a number of resources available on our website for different hepatic spheroid models. We have user guides for some of the products we talked about today, different scientific posters, and a variety of application notes. And again, all of these can be found on our web page. So with that, we'll thank you for your time, and we'll be happy to take any questions.
Thank you, Julia, and thank you, Michael and David, for your informative presentations. We will now start the live Q&A portion of our webinar. Now, to our audience, if you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Okay, we've got some great questions coming in. Let's get started. Our first question: what types of experimental factors are important to consider when setting up studies with 3D hepatic spheroids?
Okay, thanks, Christy. I think maybe I can start with this one. A couple key things to keep in mind are really the types of questions that you're trying to answer with the 3D models. What we found, and I think highlighted throughout the webinar, was the increased activity that we find, but with lower numbers of cells. And so, I think sensitivity of the assays or endpoints becomes very important here, that you have the instrumentation or assays that are sensitive enough to measure from endpoint of interest from a single spheroid or from multiples, depending on kind of the required amount of biomass, and that may take a little bit of dialing in. And related to that, and as Julia mentioned in her wrap-up, many of the assays or tissue sample preparation steps that are used or that are sort of commonly used, and we look at it comparing to 2D to 3D, don't work very well with the 3D microtissues, if you will. The tissues don't always solubilize, dissociate, et cetera, as well as just traditional monolayer cells. And so we tend to take the approach of treating them more tissue-like in in the types of either mechanical, enzymatic, or chemical dissociation that are required. So those are at least a couple things.
Thank you, David. Have you looked at primary human hepatocytes from diseased, for example, individuals with NASH tissue for ability to form and function in 3D? And do you offer these?
Okay, maybe I'll answer that one too. We have looked. So I'll start with the second question. We do not currently offer. We don't have any commercial products or available cryopreserved cells from disease models. From individuals with different types of hepatic diseases or pathophysiological conditions. What we found, excuse me, in practice is it's very difficult for us to consistently isolate high-quality cells from diseased tissue. And the heterogeneity of the tissue that comes in is pretty dramatic. So that's just been a real challenge for us thus far. We're working on it, but we don't currently have a ready-to-go solution.
Thank you. Now our next question. You showed much higher basal activity of CYP3A4 in the HepaRG spheroid cultures compared to monolayer. Can these cells be induced any further?
This is Michael. I guess I'll go ahead and answer that one. That's a great question. I have not attempted it, would be the real answer, so I'm not sure. Do you need to induce them? No. They're already orders of magnitude higher than any induced level of cytochrome P450 enzyme activity you'll get in 2D monolayers, and you don't have to worry about buying the drugs or any regulatory concerns around the drugs. But that's a really interesting question, and it's on my to-do list for sure, so hopefully I'll answer that really soon.
Thank you, Michael. Now we're almost out of time and we'll have to wrap with this final question, but I do want to let our audience know that any questions we were not able to answer today and those that come in during the on-demand period, they will be answered via the contact information you provided at the time of registration. Okay, our final question of the day. Now you mentioned that all of the lots of various species tested form spheroids. Are these platable, suspension quality, or both?
Yeah, I can take that question. It's a really great question. So far for the formation of animal hepatic spheroids, we've only tested those lots that are platable in 2D culture. We've yet to test any that are suspension grade 2D hepatocytes.
Thank you, Julia. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd also like to thank Dr. David Kuninger, Michael Millett, and Julia Tritapoe for their time today and their important research. And we'd like to thank LabRoots and our sponsor, Thermo Fisher Scientific, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now, and we hope to see you again soon. Have a great day.